Vision and muscles. So let's go ahead and get started and talk about vision. Okay, somehow, there we go. Maybe, there we go. Okay, the whole area that you can see without moving your head or eyes is known as your visual field. Now, the reality is that we're all different. I have a brother who has tunnel vision. The reason he has tunnel vision is because his eyeballs are recessed inside his skull. His skull is shaped that way. Now, this is really kind of interesting because I have four brothers and two sisters. I had two sisters. I only have one now, but I have had five siblings, and he was the only one with tunnel vision. As interesting as that was. We all kind of look alike, but for some reason his eyes are recessed inside his skull. Now my eyes are on the outside of my skull, and I've got a pretty big dip right there. So I've got a peripheral vision of about 180 degrees. I can see almost I can see a lot. My visual field is huge. So it really changed a lot of things. For one thing, since he had tunnel vision and he had brown eyes, I have blue eyes. Since he had brown eyes, when he was in Vietnam, one of the things they did, he can see really well at night because of his tunnel vision. So I don't know. Anyway, because of his tunnel vision, so he can see really well at night. I used to hate to play games with him because at night, because he could see so much better than everybody else. We used to play hide and seek and pee-wee stick down and kick the can and all kinds of interesting games at night, and he would win every time. He would win. It didn't matter um, which neighbors came over. It didn't matter which cousins were there. He won every time, and he had the ability to see things in the dark that other people couldn't see, like shadows. He was very aware of shadows, and of course, when he was in Vietnam, they utilized that. He was a night fighter, and they'd send him out on the perimeter, and he would wait there, and on the normal trails that the enemy would come in, and he would ambush them. Sometimes he'd ambush them by himself, depending on how many there were, or he'd just sneak back and tell them, well, you need to put a couple mortar rounds right here so that it'll take care of them. And he was out there by himself, as interesting as that was. And one of the other things he did was he was a tunnel rat, and he would go in without. He would have a flashlight, but he wouldn't turn it on, and he would be able to see better than most people could. Normally, the tunnels are lit to some extent. It's fairly dark in there, but most people, it's just too vague to see. But he was able to pick things out. And he came back, so came back messed up, but he came back anyway. So his visual field and my visual field completely different. He didn't see nearly, his peripheral vision wasn't good. During the day, his eyesight wasn't as good as other people's vision was. Um, he was much better at night fighting than he was at fighting during the day because he couldn't see. In football, when we played football with each other, I was faster than he was, but he was quicker than I was. So that really didn't have anything to do with his vision, except, well, it did have to do with his vision because when he tried to cover me, if I could just run straight, I was fine, but I couldn't cover him because he would cut. Just all of a sudden he would cut, and he'd be, 5, 6 feet away from me, and easy to hit with a pass. While the visual field may be wide, and an individual's visual acuity tends to be only in the middle of the field of vision. And of course, my acuity is a little bit better because I have such a large amount of peripheral vision. This is because there is a concentration of cones in the middle of your visual field, an area known as your fovea. And this is what you're seeing if you're looking directly at this building. If you're looking at it from the side, this is probably what you're seeing because you're not concentrating on it. However, even with the concentration of visual receptors in the center of the visual field, there is a spot where the optic nerve feeds out of the eye and blood vessels feed into the eye. This is known as the optic disc, and this area maintains a blind spot in an individual's visual field. Normally, the brain automatically fills in the blind spot, so you don't really notice it. Vision comes to the humans in the forms of wavelengths of light, or photons. The human visual ability lies between 400 and 700 nanometers, ultraviolet to infrared. We can't see these things, but there are animals that can see ultraviolet and infrared. Birds, for example. Bees can see in these light ranges. Other animals have ranges of vision that encompass wider spectrums of light. Bees can detect beyond the ultraviolet light, and this is one of the ways that they can follow. They can see where another bee has been just by, and you can imagine what it looks like to them. The bees are just buzzing around, and they can actually see where other bees have been. And that's how they follow a bee back to a source of pollen. A photon of light comes into our eye with select properties that we will perceive as color. The hue is the wavelength of the light. Wavelengths in the 400 to 500 range are very dark purples to blue. Wavelengths in the 500 to 600 range are greens to orange with yellow in the middle. Wavelengths in the 600 to 700 range are bright reds, darkening to black. Since most color is detected as patterns of light of different wavelengths, brightness refers to the amount of white light from the object. Saturation has to do with the amount of light of a select wavelength. If all the photons reflecting from an object were at 510 nanometers, the object would be highly saturated as green. And this is what we're talking at, 510 nanometers. The eye acts much like a camera. The eye has a transparent sac called the lens that widens and narrows to focus light on the retina. Also leading to proper focusing is the cornea, or the curved outer cover of the eye, that changes the light beam as it enters the eye. The shape of the lens is controlled by a set of muscles around the lens called the ciliary muscle. When the lens is made to widen or shorten to allow for focusing, it is known as accommodation. At birth, about 15% of males and 18% of females have severe enough eye problems to require correction. By the age of 20 until presbyopia, 48% presbyopia is when your eyes aren't as flexible as they were. This usually happens in your 40s or 50s. This is when people start wearing reading glasses if they haven't needed glasses up to that point. I am wearing reading glasses. I have never needed glasses, but I do need reading glasses now. Of course, I'm 72 years old. I started wearing reading glasses when I was in my 50s, in my relatively late 50s. So that's not bad. About 52, I guess. Okay, by age 20 until presbyopia, 48% of females and 32% of males require correction. So as you can see, females don't have as good of eyesight on the average as males do. Nearsightedness or myopia is where the eyeball is too long and the clearest focus occurs before the light hits the retina. And that is what my wife has. Farsightedness or hyperopia is where the eyeball is too short and the best focus point is past the retina. Actually, my wife has an astigmatism. An astigmatism is a misshapen cornea, elongated usually, that causes the light to pass incorrectly into the eye and cause the individual to be nearsighted. And that's what my wife is. She's nearsighted. She said her name was Acuity when she was young. So in the old days, of course, they used to arrange everybody alphabetically. So she was way back in the back of the room, and she couldn't see the board. And for this reason, teachers would write something on the board, and they'd say, "You need to copy this," and she'd be back there squinting and not being able to see it. So eventually, and they wouldn't move her up because it's the alphabet. So she had to get glasses when she was in the first grade. But it took them a while to figure all these things out. They thought there was something wrong with her other than that she couldn't see. So they got her glasses, and when she put them on, she said, trees have leaves. She just thought they were big green masses of, jello or something. Anyway, all of a sudden she could see things, and she was just excited. She still talks about it. The iris is a muscular covering under the cornea that gives the eye its color. Non-brown eyes were a mutation that appeared somewhere between 6,000 to 10,000 years ago. So those of you who have blue eyes, this is a mutation that appeared only 6 to 10,000 years ago. Yeah, so if we saw humans from, I don't know, from 20,000 years ago, they would all have brown eyes. The pupil is an opening in the iris that allows light to enter the eye. The movement of the eyeball itself is controlled by the extraocular muscles. So you can imagine the first person that came up with blue eyes, they probably killed them or something because it was such an oddity. But then it became a novelty, and potentially, of course, that's one of the reasons why there are so many blue-eyed people, which is a good question. Let's look that up. Let me go to the internet. Percentage of people in the world with blue eyes. 8% of the world's population has blue eyes. It's genetically recessive. Only about 8% have blue eyes. While blue eyes are significantly less common than brown eyes worldwide, they are frequently found from nationalities located near the Baltic Sea in northern Europe. And of course, my ancestry is English, pretty much. English and Northern European. So there you go. I had blue eyes. My dad had blue eyes. My mom, obviously, my mother produced children with blue eyes, so she was heterozygous. She had brown eyes, but she could produce blue-eyed children. Anyway, there you go. Blue eyes. Mutation. Kind of like the Wolverine, I guess. The photoreceptor cells of most animals are part of the retina on the back of the inside of the eyeball. The retina is very thin, but still contains 5 distinct layers. The first layer is the optic nerve fiber layer that is working from the inside. This is working from the inside out. So it's the optic nerve fiber layer. The second layer are the ganglion cells. The third layer are the bipolar cells. The fourth layer is the actual photoreceptor cells themselves, the rods and the cones. And the fifth layer is made up of pigmented epithelium, and that's what you can see when you shine a light into animals' eyes. You can see their eyes light up at night, including humans, of course. and that is the pigmented epithelium. So the better the eyesight at night, the brighter the light, because it catches whatever light there is. So the brighter the light, the better that they can see. You can tell if a dog is going blind because if you shine light into his eyes, it gets greener and greener and greener as his eyesight starts to fail. The rods and the cones detect light that reflects off the pigmented epithelium. The rods and the cones release neurotransmitters that synapse with the bipolar cells. The bipolar cells synapse with the ganglion cells that form the optic nerve. While most of the communication in the retina runs from outer layer to inner layer in a vertical pattern, amacrine and horizontal cells communicate with cells in the horizontal pattern. The horizontal cells connect receptor cells, amacrine cells connect the bipolar and ganglion cells. Rods and cones represent different functional systems. The scotopic system consists of all the rods in each eye, about 100 million cells. They detect color. The scotopic system is very sensitive to weak light but cannot detect color. Light is captured in the scotopic system by the photopigment rhodopsin. The scotopic system is located primarily outside the fovea. The phototopic system consists of all the cones in the eyes. Wait a minute. So the rods detect light, and the cones detect color. The phototopic system, I got it backwards, I apologize. The scotopic system has to do with just seeing light. And there's a lot of them, okay? 100 million of them. And you're just trying to detect light with your scotopic system, your rods. But your cones are the ones that detect color, and that's the photopic system. There's about 4 million in each eye. There's 100 million rods, and there's 4 million cones. This system detects color and requires relatively strong stimulation, and therefore is primarily a day vision system. The photopic system is located primarily in the fovea, but is present in other areas of the retina, but in a lower concentration. There are 3 forms of photopigments called opsins that detect the 3 color groupings. So the light is detected by rhodopsin, and the color is detected by 3 forms of opsins. The photopigment for the scotopic system is rhodopsin, as I said before. The photopigment for the photopic system is one of 3 configurations of opsins. All the photopigments in the eye are made up of 2 parts, the retinal and the opsin. Retinal is short for vitamin A aldehyde, or retinaldehyde, shortened to retinol. Retinol and opsin. Light activates the rhodopsin molecule to rapidly lose its retinol, leaving an enzymatic receptor site on the opsin molecule. The enzymatic opsin molecule combines rapidly with G-protein transducin. Transducin, in turn, acts on photodiesterase, PDE, to convert cyclic guanosine monophosphate, cyclic GMP, to 5-GMP. Cyclic GMP inhibits sodium channels closing with and with the conversion, the sodium channels will close. The loss of sodium ions depolarizes the neurons. Since we are dealing with millions of receptor cells at the same time, weak light will only hyperpolarize some of the receptors, and bright light will hyperpolarize most of the receptors. Thus, the response will always be graduated as to the amount of light that is detected. This form of response leads to sensitivity. It allows for adaptation. It allows the individual to integrate the stimulus over time. There are 2 types of bipolar cells that transmit information: off-center bipolar cells and on-center bipolar cells. On-center bipolar cells are activated when light is turned on. Off-center bipolar cells are activated when the light is turned off, so that the eye still has visual capabilities. It detects light coming in from the periphery of the visual field. On-center and off-center bipolar cells. Photoreceptor cells release the neurotransmitter glutamate. Glutamate is decreased in off-center bipolar cells when weak light is turned off. When it is weak or the light is turned off, glutamate is decreased in on-center bipolar cells when light is turned on in the middle of the visual field. Each cone in the fovea is connected to 4 types of bipolar cells. One on-center midget bipolar cell, one off-center midget bipolar cell, several on-center diffuse bipolar cells, and several off-center diffuse bipolar cells. Each midget bipolar cell connects to just one cone in the fovea. Each diffuse bipolar cell connects to several cones. When light is turned on-center bipolar cells depolarize on-center ganglion cells. When light is turned off-center bipolar cells depolarize off-center ganglion cells. Stimulus only affects the part of the retina that is in the visual field. The retina will create a mosaic of stimulation that the brain puts together as a picture. There are various ways that the eye adjusts to different light intensities. The pupil of the eye will shrink with bright light and widen with dim light. Rods and cones have different thresholds. This is range fractionation. Photoreceptors will adjust their range of reaction depending on the intensity of the light. This will give the individual visual acuity despite the quality of the illumination. Calcium is a major player in photoreceptor light adaptation. When an individual changes from one extreme light to another, it takes time to adapt to the new intensity of light. This is because the retinol and the opsin break down at higher rates in bright light and must regenerate to be available in the dark. Adaptation is much quicker when adjusting from dark to light because the retinol opsin level is not depleted. Color acuity is more accurate in the center of the visual field because the fovea is almost exclusively cones. Moving out from the fovea, the number of cones decreases and the number of rods increases. Light sensitivity is most acute about 20 degrees off center, where the concentration of rods is the greatest. So if you're looking at, you're trying to detect light at night, and there's really no reason why you should. You guys really don't go out at night, but when you're in the military and you're standing guard duty at night, usually you're standing guard duty under a light, and you really can't see out very well. But if you're in the dark, like my brother was, and you're trying to detect whether somebody's out there, of course, you know, there's moving shadows. There's always light outside, even if it's just starlight. Even on foggy nights, let's assume it's a foggy night. So what's happening? What's happening is because of the fog, the fog actually intensifies whatever light there is, as weird as that is. So you can actually see better see okay in the fog. One of the interesting things about snow is that it reflects all the light that there is, if there's any light. So when there's snow on the ground and there's no light, you can actually see fairly well because whatever light there is being reflected off the snow, as weird as that is. But of course he was in Vietnam, and there's certainly no snow in Vietnam because it was so hot. Anyway, so what are we talking about? We're talking about seeing things 20 degrees off center. So if you're trying to detect a light, and a lot of times you see light moving you go outside and there's light moving. There's light out there and it's just really dim. So the best way to look at it is not to look directly at it. If you look directly at it, you can't see it. But if you look about 20 degrees off center. You know, you just move your eyesight a little off center, you can actually see it. And this is one of the reasons why people think they see ghosts and whatnot, because there's a dim light out there that they can't see when they look directly at it. But if they look off center from it and they know what they're doing, of course they can see it. I don't know if I'm going to talk about this or not. Anyway, that's what they used to teach us when we were in the military, not to look directly at something if you're trying to detect light. About half the primary visual cortex, the V1, is devoted to the fovea. This is one of the reasons that humans are able to concentrate their spatial field on tasks close at hand, such as reading and surgery. Scotoma is a gap in a perceptual field, usually due to injury to the visual mapping area of the brain. This is in the back of your head. So if you get a blow to the back of the head, potentially you'll have a blanked out area that you can't see. But you will fill it in. That's the point. People with scotoma will often compensate for their problem by allowing part of the brain not affected to dominate the affected area. And this is known as blind sight. So this is really what they're seeing, but this is what they detect because their brain is filling in all of these missing pieces. If we look at this thing right here, this red X, eventually what will happen? Our eyesight will become fatigued, and this gray spot will go away. So it only takes about 30 seconds for that spot to get smaller and smaller and smaller. So the better eyesight you have, the faster this thing will actually go away, as weird as that sounds. But you have to concentrate on the red plus in the middle. And you can watch it actually shrink, as weird as that is. Anyway, that's a trick. Each eye is divided into two parts: the nerves of the temporal half of the visual field, the nerves of the visual half of the visual field. As the optic nerve leaves each eye, the nerves come together in an area known as the optic chiasm. At the optic chiasm, the nasal optic nerves cross to the opposite hemisphere, and the temporal optic nerves stay on the same side. After the optic chiasm, the nerve pathways are known as the optic tract. Most axons of the optic tract terminate in the lateral geniculate nucleus, the LGN, of the thalamus. From the LGN, the axons radiate into the primary visual cortex, the V1 of the occipital lobe. And that's what we were talking about before. Wait a minute, where is it? There, that's it right there. V1. So if you have vision problems, potentially what's happened is that you've had a blow to either the front of the head, you get a rebound effect. So if you get hit here, then you get a rebound effect here. If you get hit here, you get a rebound effect in the frontal lobe. Anyway, so if you get hit in the head, then potentially your eyesight will become double, or it will blur. It will be blurry. That's one of the reasons why, if we think you've had a concussion, one of the things we'll do is hold up fingers and ask you how many there are. First of all, we want you to know if you can understand what we're saying, and the other thing is that we actually want to know how good your vision is. This tells us where the blow was, as to whether you can hear us. If you can't hear us, the blow is to the side of the head. If you can't see what we're doing, then the blow was to either the front or the back of the head. The primary visual cortex is sometimes called the striate cortex because of the striations that run through the area. Visual areas of the brain not in the striate cortex are known as extrastriate cortex. Some axons of the optic tract send their axons to the superior colliculus, where the superior colliculus helps the eyes track rapid movements. And of course, this is important. If something is zipping around you, you need to know what it is and where it is. So this is the job of the superior colliculus. The LGN is arranged in 6 layers. The outer 4 are the parvocellular layers, and the inner 2 are the magnocellular layers. P cells have small receptive fields, while the M cells have larger receptor fields. M cells are sensitive to low contrast and rapid change, and these, as you can see, they're darker, they're heavier. Those are the magnocellular layers. About 10% of the retinal ganglion cells are M cells. 80% of the retinal ganglion cells are P cells. P cells require high contrast to be stimulated. P cells are relatively insensitive to rapid temporal change, such as flicker. This is one of the reasons why if you see a computer if you see a computer in a movie, a lot of times you'll see it flickering, and that's because computer screens do flicker, but we don't see that because of our P cells. P cells are sensitive to different wavelengths of light, and they therefore detect color. P cells have sustained responses. P cells have high acuity. And because they have sustained responses, the flickering of the screen is not detectable. The primary visual cortex, the V1, is the first and most abundant area of the human brain. From the V1, the stimulus is farmed out to other areas of the brain depending on what part of the eye the stimulus comes from. Areas are designated from V1 to V5. Researchers have discovered that there are 3 different kinds of cones: cones that are sensitive to short wavelengths only, cones that are sensitive to long wavelengths only, and cones that are sensitive to middle wavelengths. Of the middle wavelength cones, some are more sensitive to higher ranges of middle. Some middle range cones are more sensitive to the lower range of middle. There are twice as many long wavelength cones, reds, as middle wavelength cones, greens and yellows. Short wavelength cones are the fewest, blue and purple. And this may be one of the reasons why red is such a favorite color of everyone, because they can see it the best. Myopia is a condition where the eyeball is elongated, causing the image to focus on in front of the retina, causing a blurred image. There is some evidence that nearsightedness is caused when children look at images too close to their face, like if they were watching television close up. This can be prevented by only allowing reading in adequate light, avoiding small type, reading without glasses anytime that you can do so. Glaucoma is a group of eye conditions that damage the optic nerve, the health of which is vital for good vision. This damage is often caused by an abnormally high pressure in your eye. I have a brother with glaucoma. It's another brother, not the one with tunnel vision. He has glaucoma, and he has to put eye drops in his eyes to lessen the pressure every day. Glaucoma is one of the leading causes of blindness for people over the age of 60, and this is what happens. This is normal vision. This is early glaucoma, and this is later glaucoma, and this is what happens if you don't get it treated. And that is the end of the chapter about vision. So now we're going to talk about motor control and plasticity. I'm going to lift weights today, so this is really important. Today's my lifting day. I also lift on Mondays. Muscles cover the skeletal system and provide movement. They also provide protection for the organs that lie underneath. How muscles attach to the bones is an indication of the type of movement that they perform. Muscles only provide force when they contract, they tighten up or get smaller. And that's one of the reasons why when you're trying to show your bicep muscle, you pull your arm in. Normally, it's the muscle is long, it's straightened out, but when you pull it in, your elbow in, and make a 90-degree angle, all of a sudden your bicep will pop up. You can do the same thing with your tricep by lengthening your arm. By sticking it straight out, you can see your tricep. You can see my triceps because I work on my triceps. This is one of the areas. It's the only muscle in my body that's bigger than my son's. My son's a bodybuilder, and he's got huge biceps, but his triceps aren't as big as mine. Ha ha ha. Some muscles maintain body posture. Some muscles produce movement around a joint. Any movement that occurs in the body is performed by muscle. Muscles are connected to bone by tendons. Around a joint, there are two sets of muscle groups. When one group contracts, the other extends. These muscles are antagonists. And that's what's going on with your bicep and your tricep. You can do this. You can play with these muscles. You can make a 90-degree angle and make your bicep stick out. And then you can stick your arm straight out. Of course, your bicep kind of goes away, but then you get your tricep pops out on the back. Muscle groups that act together to perform the same act are known as synergists. Muscles are made of thousands of fibers. These muscle fibers are made up of two kinds of filaments, thick and thin filaments arranged in a regular pattern. This thick filament are made up of myosin, and the thin filaments are made up of actin. And this is what I was talking about. The bicep, you can see the bicep is contracted, and the tricep isn't. And then when you push your arm down, the bicep goes away and the tricep pops out. There are two basic types of muscles in the human body. Smooth muscles can contract in an automatic fashion without human control, regulated by autonomic nervous system. Heart muscles, muscles in the digestive tract are good examples of smooth muscles. The other type of muscles are striated muscles, primarily supporting the skeletal system. Since different muscles perform different tasks, each requires different speeds, precision, strength, and endurance. Striated muscle fibers come in two forms: fast muscle fibers and slow muscle fibers. Each movement must be quick to follow the movement of speeding objects, but only for short periods of time. Eye movement must be quick to follow the movement of speeding objects, but only for short periods of time. Extraocular muscles are fast muscle fibers. Fast muscle fibers react promptly and strongly. They fatigue rapidly. They're used when the muscle tension must change frequently. And this is one of the reasons why we can't sprint for, a mile. Can't do it. That has to be the slow muscle fiber, because fast muscle fiber fatigues very rapidly. Some slow muscle fiber have greater resistance to fatigue. They're used mainly to maintain your posture. Muscles react to stimulus coming in from motor neurons. The axon will split into many branches and activate many muscle fibers. As the axon is activated, it releases the neurotransmitter acetylcholine. Acetylcholine makes the muscle react by opening sodium and calcium channels in the muscle fibers. The ionic change allows the muscles to contract. The point where the muscle fiber and nerve structure communicates is the neuromuscular junction. Neuromuscular junctions are large and very effective. And this is one of the reasons why you need to maintain your minerals. You need to take in salt before you run. some people have drowned themselves, have killed themselves because they drink too much water before they started running, and it dilutes the body system. The muscles can't fire properly because they've diluted out so much of the sodium, and for that reason, of course, they have changed the chemical structure of their body by drinking too much water. You can poison yourself by drinking too much water. That's called hypernatremia. And people have actually gone into marathons thinking that they needed to hyperhydrate, and what they actually did was poison themselves, dilute out the chemicals in their body too much, and they actually killed themselves. And there's a neuromuscular junction right there. Each neuromuscular junction releases enough acetylcholine to not only cause one reaction, but several at the same time. One cause of muscle fatigue is due to diminished effectiveness of the neuromuscular junction. Muscles develop coordination when axon terminals migrate to specific target muscles, and acetylcholine receptors migrate to the clustered axon tips, allowing for more effective muscle reaction. Muscles react because spinal or cranial nerves send messages to the muscles through afferent neurons. Motor cells of the spinal cord are larger and faster than other motor neurons. Generally, small motor neurons activate slow muscles and are more easily excited. Large motor neurons tend to activate large muscles, which require more stimulation for activation. And now we're going to see how muscles actually work. Maybe, there we go. And are you aware? Look at these muscles.
The skeletal muscle of animals, including humans, are striated. They exist in bundles of thousands of muscle fibers. A single muscle fiber is actually a muscle cell. As you can see, muscle cells are very elongated and contain several nuclei. Packed inside the muscle cells are contractile myofibrils. Myofibrils show the striated pattern as repeating subunits called sarcomeres. When a motor neuron stimulates a muscle fiber, overlapping filaments in the sarcomeres slide along one another, and the sarcomeres shorten. The repeated pattern of the sarcomere results from the arrangement of the two major proteins within the myofibrils: myosin and actin. The myosin heads are the actual mechanism for muscle contraction. Now let's examine the process in detail. First of all, when a motor neuron stimulates a muscle fiber, calcium ions are released from the sarcoplasmic reticulum. The ions force the tropomyosin to displace and expose myosin binding sites on the actin. An ATP molecule is then hydrolyzed, and the myosin head forms a cross bridge. When the ADP is replaced by a new molecule of ATP, the myosin head detaches from actin and goes back to its original state. And that's pretty much the whole process. And one more thing. Each myosin head attaches to and releases from actin independently, not in unison. Otherwise, the myosin heads would all release at once, and the whole muscle would go to a relaxed state. And that wouldn't be good.
Of course, the funny thing is that while you're lifting weights, you hear this music all the time. Just kidding. Okay, anyway, that's how muscles work. The body must maintain control over the position of its parts. This knowledge of position and state of its muscles and limbs is called proprioception. This knowledge is maintained by select receptors that report muscle and joint positioning. Muscle spindles lie parallel to muscle fibers. Golgi tendon organs report on the position of the joints and are situated with one end attached to the tendon and one end attached to the muscles. Muscle spindles are located in many areas of your skeletal muscular fiber. Muscle spindles are actually spindle-shaped, and it consists of an afferent element and an efferent element. Their primary function is to maintain their posture and tell you when you have been in a position too long. When a muscle fiber is activated, the muscle spindle reports rate of change. Rate of change refers to the change that the fiber has gone through to be able to function. The muscle spindle also reports force, the force required to maintain contraction. Muscle spindles have two types of receptor endings. Primary sensory endings wrapped around the middle of the fiber are activated during the initial phase of contraction and then adapt to a lower discharge rate. Secondary sensory endings, found near the smaller end of the fiber, are not activated at first, but only after prolonged use. When a muscle fiber is activated, the muscle spindle reports rate of change. Rate of change refers to change that the fiber has gone through to be able to function. The muscle spindle also reports force, the force required to maintain contraction. Muscle spindles have two types of receptors. We've already done this. I went backwards. Muscle spindles are informed of impending action through two special motoneurons. Gamma motoneurons, or gamma afferents, inform of change of position from the initial movement. They comprises about 30% of the efferent fibers, and alpha motoneurons, performing initial movement, 70% of the efferent fibers. While individual muscle movement is monitored by the muscle spindle, muscle contraction is monitored by the Golgi tendon organ. Golgi tendon organs are able to monitor muscle and joint movement because joint movement is controlled by tendons connected to muscles. Golgi tendon organs, there you go, right here. Golgi tendon organs are somewhat insensitive to passive muscle stretch because they are connected to the elastic component of the tendon, and it is only activated under heavy loads. The Golgi tendon organs detect overloads that threaten to tear muscles and tendons. So if you've ever tried to lift something that was too heavy, then it was probably your Golgi tendon organ that told you, you better not do this, or you're going to tear your muscles. Activating the Golgi tendon organs inhibits the motoneurons supplying the muscles that pull on the tendon, and thus by relaxing the tension prevents mechanical damages. Muscle building. Let's see if this one's still active. There we go.
Muscle building. The key to building muscle is protein synthesis and cellular repair. Skeletal muscle is made up of long fibrous chains containing proteins. Bundles of thousands of fibers make up the muscle itself. When an athlete engages in a heavy workout, this structure is damaged, causing micro tears in the muscle fibers. The body naturally repairs this damage by mending the torn fibers. The result is a muscle that's larger and stronger than before. And with each additional workout, the athlete adds bulk, strength, and speed.
There you go. That's how it works. And that's one of the reasons why I'm going to go lift today. Not so that my muscles get bigger. My muscles, I lift the same amount every time, but just to maintain my muscle mass. At my age, muscles atrophy fairly readily because, well, I'm old. So if I ever stop, then they will go away, as strange as that may seem. So this is really kind of interesting. What in the world's happening here? Anyway, let's go to this one and see if that one still works. There we go.
Muscles. We have over 600 of them. They make up between 1/3 and 1/2 of our body weight. And along with connective tissue, they bind us together, hold us up, and help us move. And whether or not bodybuilding is your hobby, muscles need your constant attention because the way you treat them on a daily basis determines whether they will wither or grow. Say you're standing in front of a door, ready to pull it open. Your brain and muscles are perfectly poised to help you achieve this goal. First, your brain sends a signal to motor neurons inside your arm. When they receive this message, they fire, causing muscles to contract and relax, which pull on the bones in your arm and generate the needed movement. The bigger the challenge becomes, the bigger the brain's signal grows, and the more motor units it rallies to help you achieve your task. But what if the door is made of solid iron? At this point, your arm muscles alone won't be able to generate enough tension to pull it open, so your brain appeals to other muscles for help. You plant your feet, tighten your belly, and tense your back, generating enough force to yank it open. Your nervous system has just leveraged the resources you already have, other muscles, to meet the demand. While all this is happening, your muscle fibers undergo another kind of cellular change. As you expose them to stress, they experience microscopic damage, which in this context is a good thing. In response, the injured cells release inflammatory molecules called cytokines that activate the immune system to repair the injury. This is when the muscle building magic happens. The greater the damage to the muscle tissue, the more your body will need to repair itself. The resulting cycle of damage and repair eventually makes muscles bigger and stronger as they adapt to progressively greater demands. Since our bodies have already adapted to most everyday activities, those generally don't produce enough stress to stimulate new muscle growth. So to build new muscle, a process called hypertrophy, our cells need to be exposed to higher workloads than they are used to. In fact, if you don't continuously expose your muscles to some resistance, they will shrink, a process known as muscular atrophy. In contrast, exposing the muscle to a high degree of tension, especially while the muscle is lengthening, also called an eccentric contraction, generates effective conditions for new growth. However, muscles rely on more than just activity to grow. Without proper nutrition, hormones, and rest, your body would never be able to repair damaged muscle fibers. Protein in our diet preserves muscle mass by providing the building blocks for new tissue in the form of amino acids. Adequate protein intake, along with naturally occurring hormones like insulin-like growth factor and testosterone, help shift the body into a state where tissue is repaired and grown. This vital repair process mainly occurs when we're resting, especially at night while sleeping. Gender and age affect this repair mechanism, which is why young men with more testosterone have a leg up in the muscle building game. Genetic factors also play a role in one's ability to grow muscle. Some people have more robust immune reactions to muscle damage and are better able to repair and replace damaged muscle fibers, increasing their muscle building potential. The body responds to the demands you place on it. If you tear your muscles up, eat right, rest, and repeat, you'll create the conditions to make your muscles as big and strong as possible. It is with muscles as it is with life. Meaningful growth requires challenge and stress.
Okay, one of the reasons that people who use steroids are able to make massive muscles is because what the steroids do, steroids are androgens, which are synthetic, they're synthetic, they're synthetic testosterone. And so what happens is they're able to repair their muscles faster. And because they're able to repair their muscles faster, they can go back into the gym and lift heavy weights again. And that breaks them down, and then they repair faster. Because they're repairing faster, the muscles build up a lot faster. So if you've ever been around somebody that's taking steroids and they were lifting weights, one of the things that you probably noticed was they got big fast. And that's one of the things that happens. So when you're talking about bodybuilders, a lot of times you're talking about did they do it naturally or did they use steroids? If they use steroids, of course, it's a lot easier to bulk up faster. If they did it naturally, of course, it takes a little bit longer. It takes longer, about twice, maybe 3 times as long. So that's one of the reasons why people take steroids, because it repairs their muscles faster after only 1 or 2 days. They can lift. I lift twice a week. Your muscles will start atrophying after about 3 days. But if you're taking steroids, you can lift almost every day. You can probably lift every day. One of the things that happens if you go to the gym and you haven't been working out and you lift weights, you'll probably be really sore for about a week, and that's because your muscles have torn down. The other reason is because one of the things that you need to do when you lift weights is consume liquids. Because the liquids will help repair the muscles more readily. If you are dehydrated when you're lifting weights, when you're doing something, then you'll get sore a lot faster because your muscles will break down, and now they'll swell up because they don't have enough fluid. They're dehydrated. Now the damage is double. So what will happen is it'll take you longer to repair your muscles, unless you consume liquids while you're lifting, and that's what I do. How do I do that? I'll take about a quart of liquid, and I'll drink about half of it while I'm lifting, and I lift for about just over an hour. So you need to take in some liquid, and I rarely have sore muscles. That and the fact that I really don't lift that heavy of weights. I've been lifting the same amount of weights for the last 30 years or so. Okay. Anyway, not important, or this is really important, so things to think about and listen to. There are 2 reflexes that are maintained by the spinal cord that are automatic and are not mediated by the brain. The flexion reflex is activated when pain is applied to the leg. The muscles will contract, moving the affected part away from the stimulus. And of course, these are known as reflexes. Let me show you some of the other reflexes. Oh, here we go.
The Hulk is an anthropomorphic exoskeleton which mimics the human form. It provides extra support that enables a person to carry more weight than they normally could. Exoskeleton technologies are considered part of the robotics field. If you want to think about them as wearable robots, that's pretty accurate. They sense what the user wants to do, where the user wants to go, and mimics the motion. We like to call these mantonomous systems. The exoskeleton is primarily composed of titanium components, which are both lightweight and provide the strength needed to carry the load. A microprocessor takes in readings from the sensors that are throughout the structure and calculates where the user wants to move, and then commands the actual hydraulic system to actuate the joints to provide the motion. You can walk, you can run, you can bend, you can crawl, you can leap. It does not impede your range of motion whatsoever. This technology could be a real benefit to the military. Just imagine you're a soldier operating at 6,000 feet in the Afghan mountains and being asked to take 120 pounds up in that level of thin air. How exhausted you would be once you got there. An exoskeleton provides the ability to carry that weight the same distance, but to have energy left to execute the mission once you're there. We've also looked at attachments that allow users to carry weight not only on their back, but also on the front. The exoskeleton will fit anybody between 5'4" and 6'2". The current system weighs 53 pounds, but it's pretty much unnoticeable to the user because what the technology does is it even transfers its own weight to the ground, in addition to the weight you're carrying. And we're taking the load off the soldier's back, and we're going to help prevent injuries that occur from carrying heavy loads and repetitive lifting tasks. The best part about working on this technology is that it gives us a glimpse into the art of the possible.
Hulk. They don't use these with the military. They're having problems with the joints, not the joints of the of the Hulk, but the joints of the person. It's over extending the joints. Anyway, what was I going to say? Oh, I was talking about hydration. Yeah, you need to hydrate. Keep things hydrated. In the need, well, see, and another problem that I have is that I sweat a lot. I'm from where it's cold, so when I exercise a lot, I sweat a lot. I've always been a heavy sweater, so I lose a lot of water, and I need to replace the water. So that's another reason why I need to hydrate or stay hydrated. In the knee jerk response, the quadricep muscle that is contracted to allow the knee to bend is informed of tendon pressure that makes the muscle react by stretching. Thus the stretch reflex. And this is, of course, the knee jerk reflex. If you've ever hit this tendon in your knee, it made your foot kick up. The primary lateral cortex controls all the muscle movement in the body. The hands and lips utilize the biggest portion of the area compared to the percent of the body that they represent. And of course, this is a picture of our that portion of our brain. Our hands are very sensitive. Our lips are very sensitive. Our ears. The M1 communicates with the rest of the body through the pyramidal system. The pyramidal system is a triangular-shaped bundle of nerves that runs from the brain through the medulla and into the spinal cord. Lesions to this system will result in the inability to move select joints and limbs. Those neurons that run from the M1 to the muscles that are not in the pyramidal system are said to be in the extrapyramidal system. These neurons control the spinal reflexes. And now we're going to look at somebody. Yes, we are. Okay, they're going to do reflexes.
The lower body reflexes will be performed in the same manner as the muscle strength exams were performed. We're going to test one side and then quickly test the other side and compare side-to-side responses. We've got 5 reflexes that we're going to go over, one being the knee reflex, one the Achilles reflex, and then the adductor reflex, the internal and external hamstrings. The first one will be the knee reflex. I'm going to have the patient close their eyes, strike the tendon. We got a good response. I want you to keep your eyes closed. Strike it again. Got another good response. Now the Achilles reflex. I'm going to just slightly dorsiflex the foot by reaching underneath the foot and letting her rest the foot on my hand. And then I'm going to reach back and I'm going to tap the Achilles tendon. And I got a real nice plantar flexion of the foot. Comparing side to side, I have the patient lay face down. Now we can do the adductor, internal and external hamstring all in this position. If I'm going to test the adductor, I'm going to have the patient medially rotate the leg. I find the adductor tendon along the medial side of the leg. I've located it, and I'm going to strike over the tendon, and I get a slight contraction of the adductor. For the internal and external hamstring, I just have the patient lay flat. I locate the tendon by slightly having the patient bend their knee against resistance, bring the leg up. I'm going to push down. And the tendons pop out quite easily. I've got them, you can relax the leg. I've got the tendon located, the medial hamstring tendon. I'm going to strike the tendon, and I'm watching for contraction and slight flexion of the knee. On the lateral hamstring, I locate the tendon, and I'm going to do the same thing, strike the tendon, and watch for contraction of the hamstring as well as slight flexion of the knee.
Here we go. Oops. There we go. The extrapyramidal system communicates with the spinal cord through two tracts, the reticulospinal tract and the rubrospinal tract. The extrapyramidal system not only handles spinal reflexes, but muscles responsible for breathing and muscles that control the head and the neck. And that is the end of the chapter. So I have to go lift. I'll talk to you guys later. I'm going to go do all this stuff and probably potentially injure myself. So hopefully not.